All right, so remember, apoptosis is going to be our programmed cell death. Okay, remember, we distinguish this from necrosis by the fact that we usually don't have inflammation when we undergo apoptosis. So typically there's no inflammation. Usually this is the cell basically kind of shutting down for whatever reason. Usually there's some signal that causes this to happen. And there's two major pathways that this is going to go through. So there's an extrinsic pathway we call the death receptor pathway, and there's an intrinsic pathway or the mitochondrial pathway. All right, so I'm actually going to kind of skip this general principle section. We're going to come back to it in a minute, but I just want to talk about this extrinsic pathway first, actually. So the extrinsic pathway is kind of like it says. There's a signal that's essentially coming to the cell from the outside. Okay, so there's some extrinsic signal that's coming to the cell, and that signal is going to tell the cell to undergo apoptosis. Okay, so just for an example of this, let's just say that there's some virus that's infected this cell. Okay, so there's some virus that's infected the cell, and it's trying to hijack this cell's machinery. Now, normally, what will happen is these cells, right, all of our normal cells are going to have MHC1 on the surface of their cells, right? All nucleated cells will have this. And what these MHC1 proteins will do is they'll display some of these viral antigens to kind of tell the body that, hey, this cell is infected with something, right, intracellularly. And usually this MHC1 will then send that signal to a CD8 positive T cell, right? So remember your rule of 8s from immunology. And this CD8 positive T cell could then send a signal to essentially tell the cell, hey, you need to undergo apoptosis because you're infected, and we don't want this virus to use the cell to replicate its machinery. Okay, now some viruses potentially could downregulate MHC1 expression, right? They could downregulate this, and then what the body would have was no MHC1 expression on its surface. And this would be recognized by a natural killer cell. Okay, so natural killer cells are essentially looking for cells that lack MHC1 expression, and they will destroy those cells with similar mechanisms. Okay, so the point is that when a cell is infected with a virus, or if it's a cancerous cell that's displaying cancerous antigens, we have signals that can come in from the outside to essentially help these cells undergo apoptosis before things get out of control. Okay, so that's the general idea of this extrinsic pathway and why it exists. Now, TNF-alpha we know is classically associated with the inflammatory response. And this is one substance that potentially can set off this pathway. So TNF-alpha can bind to a tumor necrosis factor receptor here on the cell surface, and this can set off a cascade. So once this binding occurs, we get initiator caspases. So let's talk about these really quickly. Caspases are going to be cysteine aspartate proteases that are essentially going to start to set off a cascade that leads to these executioner caspases, right, just like we saw, to eventually cause apoptosis. Okay, and that's really the extent of what you have to know, right? So in this extrinsic pathway, we're essentially getting some signal from the outside, in this case by TNF-alpha, cause these initiator caspases to then activate and eventually cause destruction of the cell. Now, the other option here is we have this Fas and Fas ligand that can bind to a Fas receptor. So in a similar process, it can cause the extrinsic pathway to be activated. And these receptors are sometimes called the death receptors, classically the Fas, or sometimes referred to as the CD95 receptor, right? And then you also have the TNF receptor that we talked about, leading to, again, receptor ligand complex, right? This is the receptor, here's the ligand, they complex, and you get activation of caspases. So that's kind of the classic pathway. Now, the thing you have to understand about the Fas pathway, right, this one over here, is that this is that same pathway. If you remember back in the immunology section, we talked about, there's positive selection and negative selection, right, occurring in the thymus when we have T cell development. We have to make sure our T cells that are mature and go out into the body are not going to attack self. Right? So that's what negative selection is doing. We got to make sure that our own T cells don't bind too tightly to our own antigens. Now, if they bind too tightly to our own antigens in this process of negative selection, then what happens is the Fas ligand will bind to this Fas receptor, and it will cause these T cells that bind too tightly to be destroyed by going through this extrinsic death receptor pathway. Now, there are a couple exceptions to this, right? The exceptions that we talked about in the immunology section were these regulator T cells, the Tregs. But in general, this is the process that typically happens. So we end up making this association with Fas and negative selection. So the reason that I even go through that with you is because sometimes you'll get questions and they'll say, okay, well, what happens if there was a mutation with the Fas receptor? Let's say there was a mutation at the Fas receptor. What would be a classic presentation of a patient that has a mutation there? And you got to think up a little bit, right? You know that the Fas receptor is associated with the extrinsic pathway leading to apoptosis, and it's primarily utilized by negative selection for T cells. cells. So if I knock this receptor out, right, I might have a lot of T cells that go out into the bloodstream and out into the body that are going to be attacking self because I have a defective process of negative selection at that point. Okay, so I might have a lot of autoimmune reactions potentially if I had a mutation at the FAST receptor. So this is where you have to kind of think through what's happening and make sense of it. All right, and then the last thing on here that we kind of touched on is that these same pathways are used by CD8 positive cytotoxic T cells, right, which can bind to MHC1, for example, if the cell was infected with a virus, and then these cytotoxic T cells can release perforin, granzyme B, a lot of the stuff we talked about already in the immunology videos, and those substances that the cytotoxic T cell releases can then also go on to activate the caspases. Okay, so we have our TNF alpha, we have our FAST ligand, and then we have perforins, which can essentially put holes in the membrane, and then our granzyme B, which can go on to assist in the activation of caspases. All of this happening from an extrinsic or external source. Let's clean this up a little bit, and let's talk about the intrinsic pathway here. So in the intrinsic pathway, sometimes called the mitochondrial pathway, okay, in this pathway, usually there's some damage intrinsic to the cell, right? So there's something that's happening in the cell, maybe a genetic defect or a mutation, right? Or DNA damage from radiation that's essentially causing an activation of a bunch of different factors that ultimately leads to the same thing. We have the activation of these executioner caspases that causes apoptosis. Okay, so let's kind of walk through this. Let's just say there's some, radiation or damage to the DNA that is going to get picked up, and that's going to activate this thing right here, P53. Okay, so we're going to have activation of P53. And P53 is particularly high yield. P53 is a tumor suppressor gene, and we're going to talk about what that means as we go on through this series. I don't want to get off track, but it's a tumor suppressor gene that is essentially acting to prevent unregulated cell activation. So when there's damage, P53 gets activated and then goes downstream to stimulate some of these different factors here, which are called BAX, BAD, and BAC. Now, the thing that I want you to understand here is that the only one that I would really, if I had to remember one of these, I remember BAX. That's the really high yield one you have to remember. And these factors will eventually lead to the release of cytochrome C from the mitochondria. Okay, so you can see that here. Cytochrome C is going from the mitochondria out into the cytosol, and the cytochrome C is what signals these caspases to eventually lead to apoptosis. So quite simply, there's really three main factors you have to know: P53, BAX, and cytochrome C. Okay, and again, cytochrome C, once it gets into the cytosol, it's going to complex with this APAF1 to form an apoptosome, which causes the activation of caspases. But again, the big three that I remember: P53, BAX, cytochrome C. Now, what you want to understand here is that BAX, BAD, and BAC are all pro-apoptotic, meaning that they're going to cause increased mitochondrial permeability. If there's more permeability, there's a better chance for cytochrome C to leak out into the cytosol. And this is exactly what we said when we talked about cell injury. We said you can get mitochondrial permeability. Once that starts to happen, you start to get into this irreversible state because you get cytochrome C leaking out into the cytosol, which eventually will lead to caspase activation. Now, on the other side of this, though, there are factors that kind of keep this in check. And some of those are going to be BCL2 and BCL-XL. The high yield one you really, really want to remember is going to be BCL2. And this is a very high yield factor. Now, this is an anti-apoptotic factor. What's really tricky is these factors, which are pro-apoptotic, are actually in the BCL2 family, but they're pro-apoptotic, whereas BCL2 in and of itself is anti-apoptotic. So don't get confused by that. But when you see BCL2, you're thinking anti-apoptotic. In other words, it's preventing apoptosis. How does it do that? Well, it keeps the mitochondrial membrane really strong. So we're keeping the cytochrome C in the mitochondria. So that's why you see this inhibitory sign here. So BCL2 keeps this membrane nice and strong so that this cell does not undergo apoptosis. Well, here's the problem with that. If for whatever reason, let's just say that I have way too much BCL2 being produced for whatever reason, right? If I have way too much BCL2 being produced, what's going to happen is I'm not going to be able to get any apoptosis, right? I can't get apoptosis because no matter what happens, this BCL2 is going to keep that cytochrome C in the mitochondria because I have so much BCL2. Okay, so cells that are damaged, right, that have some genetic damage, those cells can't undergo apoptosis. So they're going to continue to replicate daughter cells that now share this genetic defect. And this is precisely how we start to get different types of cancers that form. And a really classic example of this, where we see BCL2 overexpression, is going to be in these 14-18 translocations, which is actually very classic for follicular lymphoma. So essentially what's happening is the BCL gene, the BCL2 gene, which is on chromosome 18, it gets translocated to a more highly transcribed area on chromosome 14. So now you have, this area on chromosome 14 transcribing a lot of BCL2. It's producing a ton of BCL2, and all this BCL2 that's getting produced is essentially preventing any cytochrome C from leaving the cells, and we end up with this lymphoma. And we have a whole video on follicular lymphoma, so I don't want to go into too much detail here. You can also see this with CLL, but again, like I said, we'll talk about all of this coming up. And the other thing here that you can sometimes see that's kind of interlinked with this pathway is the HPV, human papillomavirus, particularly strains 16 and 18. Now there's two major proteins. There's E7, which interacts a little bit with RB, and we'll talk about that coming up. But the E6 protein actually causes inactivation of P53. So again, same concept. If you can imagine inactivation of P53, right, what's that going to do? Well, you'll never get these high levels of Baxin, some of these other pro-apoptotic proteins to cause cytochrome C release. So it's the same concept. The cytochrome C is not going to come out of the mitochondria, and so you're not going to get apoptosis. Okay, so it's the same concept. And we see this in the cervical tissue, right, from these infections. And so you get proliferation of abnormal cervical tissue eventually, right, because you can never undergo apoptosis when there's cellular damage. That's essentially what the virus is causing to happen, all doing this by inactivating P53. The fast receptor or fast ligand defect is going to cause an increase in self-reactive lymphocytes, right? We talked about that because this is what we use for negative selection. So you can get autoimmune phenomena or autoimmune lymphoproliferative syndromes where we have T cells that are essentially attacking a cell. Finally, the last one on here is an 8-14 translocation where we again are moving a factor from chromosome 8 to chromosome 14. This spot on chromosome 14, like I said here, with BCL2, it's very highly transcribed. Okay, so we take this C-Myc protein, which we haven't really talked about yet, and again, we're going to put it on chromosome 14, so we're going to get a ton of this C-Myc activation. This area, by the way, on chromosome 14 is an area where we have immunoglobulin heavy chains being transcribed. So it's a very frequently transcribed area. Okay, so whatever I put there, if there's any translocation where I put something there like BCL2 gene or the C-Myc gene, I'm going to get a lot of transcription for that. And again, the C-Myc gene is going to be a pro-proliferative transcription factor. Okay, and so you can also see this very classically actually with Burkitt lymphoma. But this is just to give you an idea how this all kind of comes together with some of the things that you'll probably see in some of these other sections.